Have you ever just stopped and wondered, what does it actually take to make a cell? I mean, really think about it. How do you go from a bunch of simple, non-living chemicals to, well, to the fundamental unit of life itself? It's genuinely one of the biggest questions out there. So, let's dive in and try to unpack the recipe for life.
Just look at this thing. It's not some piece of abstract art. This is a single sensory neuron. It's like a tiny explosion of complexity, an unbelievably intricate little machine. And everything alive, from you and me to the smallest bacteria, is built from these incredible units. So where in the world does something this complex even begin? How would you even start to build one from scratch?
Well, to figure that out, we have to ask an even more basic question. What are the fundamental ingredients for life? You know, if we were putting together a cosmic cookbook, what would be on that list? It turns out, when you boil it all down, you really only need 3 key things.
All right, first up on our list, chemistry. It's pretty obvious, right? Before you can build a house, you need the raw materials, the bricks, the wood, the nails. For a cell, you need molecules. But where did the first organic building blocks even come from on a young, totally lifeless Earth?
This brings us to a huge, and I mean huge, idea: abiogenesis. All it means is the theory that under just the right conditions, life can emerge from non-living stuff. We're not talking about magic here. This is pure chemistry. And back in the 1950s, 2 scientists, Stanley Miller and Miller-Urey, had a brilliant idea. What if they tried to replicate that primordial chemistry right there in the lab?
And this beautifully simple setup is what they came up with. This is the Miller-Urey experiment. Their goal was pretty straightforward. Could they recreate the primordial soup of early Earth right there in a glass flask? Could the basic chemical ingredients for life just form, all on their own, with a little bit of energy?
And what's so cool is how elegantly simple their approach was. They basically bottled the atmosphere of the early Earth, simple gases, a spark to act like lightning, and a little bit of heat to create a mini ocean and rain cycle. It was an incredibly bold attempt to see if chemistry, if you just left it alone, would start to build something interesting. So they set it up and they let it run for a week.
And the result was an absolute bombshell. When they analyzed the water at the bottom, they found it was swimming with amino acids. These are the building blocks of proteins, which are the molecular machines that do almost all the work inside a cell. This was the first real proof that the jump from a dead planet to the raw materials of life wasn't some miracle. It was just plausible chemistry. These things could have formed naturally all over the early planet, just waiting for the next step.
Okay, so we've got our building blocks. We have our bricks. But a pile of bricks doesn't just automatically become a house, right? You need a blueprint. You need a set of instructions. And for life, that is our second crucial ingredient: information.
And this is how that information flows. This diagram, it looks complicated, but the idea is simple. It's called the central dogma of molecular biology, and it's basically the chain of command for all life. You start with DNA. That's the master blueprint, the permanent code. That code gets transcribed into a temporary message, which is RNA. And then that message gets translated to build a specific protein. So DNA to RNA to protein. That's the operating system for life.
The power of this information system is, well, it's staggering. A single stem cell, which has just one master blueprint, can follow different parts of those instructions to become a muscle cell or a neuron or a fat cell, every single specialized cell type in your body. It all just depends on which part of the code gets read.
Okay, so if DNA is the master blueprint for pretty much all cells, is it really in all of them? Let's do a quick quiz. Which one of these super important cell types in your body actually throws its DNA in the trash once it's fully grown up? Is it a brain cell, a red blood cell, a bone cell, or an eye cell? Take a second, think about it.
If you guessed B, red blood cells, you got it. That seems totally backwards, doesn't it? A cell that literally throws away its own instruction manual? But believe it or not, there's a fantastic reason why it does this.
So, this process is called enucleation, and by kicking out their nucleus, which is where the DNA lives, red blood cells free up a ton of internal space. And why would they do that? To pack in as much hemoglobin as physically possible. That's the protein that carries oxygen. The trade-off, of course, is that they can't repair themselves or divide, but for their one super specialized job, it's a brilliant sacrifice.
So, let's recap. We've got our chemistry, we've got our information. What are we missing? Well, you need a place for it all to happen. You need a workshop. And that brings us to our third and final ingredient, compartments.
See, a cell isn't just a squishy bag of chemicals all mixed together. It's more like a highly organized city with different districts or compartments that are usually separated by membranes. These create little specialized microenvironments where specific chemical reactions can happen without messing up all the others. It's how you bring order to all that chemical chaos.
And in a weird way, this was the very first thing we ever even noticed about cells. Way back in 1665, a guy named Robert Hooke pointed his pretty basic microscope at a thin slice of cork, and what he saw was this honeycomb of tiny, empty boxes. They reminded him of the small rooms in a monastery, which in Latin were called cella, and so he called them cells. He was literally looking at the compartments.
So there we have it, our 3 ingredients: chemical building blocks, genetic information, and compartments to keep it all organized. And when scientists started to piece these ideas together, it led to the single most foundational theory in all of biology. So after Hooke's discovery, it actually took almost 200 years before 2 other scientists, Schleiden and Schwann, proposed that all living things, not just cork, are made of cells, and that the cell is the basic unit of life. But there is still one piece missing. Where do new cells come from? Well, in 1855, a physician named Rudolf Virchow finally provided the answer.
He stated it in this absolutely elegant and powerful Latin phrase, omnis cellula e cellula,
Which means, very simply, all cells arise from pre-existing cells. No more ideas about spontaneous generation. Life only comes from life. This was the last piece of the puzzle, and it clicked everything into place.
And that right there, that gives us the whole shebang, the complete cell theory, which is the bedrock of modern biology. 1. Everything alive is made of cells. 2. The cell is the most basic building block. And 3. They don't just pop out of nowhere. New cells have to come from old cells. It's so simple, but so powerful. And it all ties back to our 3 ingredients.
Okay, so we've got our recipe. We have a pretty good idea of what a cell is, but that just opens the door to a final, even bigger question. When you manage to get chemistry, information, and compartments all together in just the right way, what does the life that results from that actually do?
Well, it does all of this. It creates something that's highly complex and organized. It runs on a genetic program, it can reproduce, use energy, carry out chemical reactions, respond to the world around it, regulate itself, and, this is the big one, it evolves. All of that emerges from just those 3 simple ingredients, which it really makes you wonder, doesn't it? Is life just what chemistry does when it gets organized enough to start evolving? That's a pretty profound question to leave you with.